Hello everyone, welcome to Illumina NGS or Next Generation Sequencing. This is one of the most complicated sequencing methods and also one of the most popular ones. Now, I'm probably sure that you have watched other videos on Illumina as well, and it is very hard to understand. I get that. So in this video, I'm going to make everything crystal clear for you step by step. This video is going to be a bit long, but bear with me. I think you will be able to understand everything because not only will I cover Illumina sequencing, but I'm going to cover the background knowledge that are required to understand the sequencing method. So without further ado, let's dive in. So first of all, let's understand a high-level overview of sequencing process. So suppose you have some human, maybe it's me, and this is my DNA, right? And I want to sequence the DNA. So what does sequencing mean? Sequencing means to be able to read the whole DNA from start to end, right? And this is not possible at a stretch because current tools are not efficient enough or they are not accurate enough to actually read the whole DNA at one go, right? So we need to fragment the DNA. Now, if you have a very small amount of DNA, then you need to clone it multiple times. There are procedures for that. So after doing that, you need to fragment the DNA into small pieces, and different sequencing methods actually do it differently. So now your main task in the sequencing is to actually understand, which fragment, these are small fragments, contain which string. So what is the string here? What is the string here? What is the ATCG string here? So that is what we need to find out using the sequencing platform. It can be some sequencing platform. For example, it can be Illumina sequencing. I don't know. So finally, now that we know the strings, and since I'm a human, so I will need to align my fragments to an ideal human reference genome. So that is what is being done here. So all the fragments are being aligned to the reference human genome. And after aligning, you can see that in the same position, there are multiple fragments. And so we can very easily take their consensus and find the actual string that should be my own DNA. That is the entire process for sequencing, but we are mainly going to focus on the cloning, the fragmentation, and mostly the sequence reading part, because that is at the core of the sequencing. The last part that I talked about here, the alignment part, that is not really part of the sequencing machine. Rather, it is done using some library. Maybe, I can give you some examples like Bowtie, BWA. These libraries are actually used to do these things. So it's not part of the actual sequencing process. Okay, so let's talk about some background knowledge before we move on to Illumina sequencing. So I think you all probably know this, but just for those who don't know, DNA is double-stranded. So we have this strand from 5' to 3' end. So this one is called the forward strand. And then there is another one which starts here 5' end and ends right here, which is at the 3' end. This is called the reverse strand. So these two strands, they sort of join with each other using what? Using what is called complementary bases. So what is complementary base? So you can see that adenine or A always pairs with thymine or T. Then cytosine or C always pairs with guanine or G. That is what complementary strand is all about. So I'll give you an example here. So for example, you can see right here that there is a string, a DNA string A, C, A, G, G, T. So if we know this string, then we can already know what will be the complementary string, because then A's complementary base is T, then C's complementary base is G, G's complementary base is C, and so on and so forth. So if we know one string, we automatically know the other one. So that is the beauty of DNA. The next thing we need to understand is the replication process of DNA, because in Illumina sequencing, we sort of use this synthesis process, the DNA making process, in order to understand or read the sequences of the fragment. So let me just briefly talk about this. So you can see that here we have the original double-stranded DNA, and it's broken up into two single strands, right? Two single strands. So there is this yellow thing which is called the DNA polymerase enzyme. So this enzyme synthesizes the DNA. So if you have a single strand and you have this polymerase enzyme and you have some specific sequence which is called primer present somewhere, then the DNA polymerase starts synthesizing from there using the complementary bases. So you can see that from the single mother DNA, we will ultimately form two children DNA, right, which are both double-stranded. So that is what is known as DNA replication. So this process will also come in handy when we talk about the Illumina sequencing. So let's now talk about Illumina sequencing. The first step of Illumina sequencing is to do library preparation. So let's talk about why we exactly do that. So we need the fragments to bind to a flow cell. There is something called flow cell, which I will show you later on. So that's one purpose. And after binding to flow cell, there is something called bridge amplification. So we don't really work with the given fragments, rather we amplify them into small clusters, and I will show you why. So the library preparation step also facilitates this. And finally, as I said before, we sort of monitored the DNA synthesis process using the DNA polymerase enzyme and try to figure out the actual sequence. So the synthesis process requires some specific primers to start. So those are also part of the library preparation step. So let's look at the flow chart of this. So we have this input DNA. We fragment them using a technique called shotgun fragmentation, and understand that these fragments are normally very short, less than 400 or 500 base pair typically. Then we attach some sequencing adapters with it. I will talk about these adapters right now shortly. And finally, if you think that there aren't enough fragments right here, so you can do something called PCR or polymerase chain reaction amplification, which will basically double or triple, or if, I mean, depending on how much amplification you want, you can double or triple or quadruple the amount of fragments using the PCR amplification. So now let's look at the adapter itself. The adapter, like what does it look like? So it looks something like this. So let me discuss it very briefly now. But as we move on with this tutorial, everything will be clearer gradually. So first of all, we have the P5 and the P7. I mean, okay, let's talk about something else first. So first of all, this middle part. So this is the fragment, the actual fragment. So you can see the middle blue part. So this is that part, the double stranded fragment. So the two ends, the P5 and P7, are flow cell binders. So on the flow cell of the Illumina sequencer machine, you have the complementary sequence of the P5 and the complementary sequence of the P7. So this complementary sequences on the flow cell helps the strand, the fragment to bind to the flow cell. That is why we have the P5 and P7. Then we have two indexes, index 1 and index 2. The other thing is Illumina sequencing is very efficient. So in the same sequencing platform, you can actually provide multiple patient DNA. So suppose you have like 3 patients and you want to sequence them together, so you can provide all their fragments together on the sequencing machine. Now, how do you keep track which fragment belongs to which patient? And that is where indexing comes into play. So you just look at the index of the patient and you understand that, okay, this fragment belongs to this patient. And finally, we have the synthesis primer. So as I said before, we are going to need some small sequence, which is called primer, for the DNA polymerase to start the synthesis process. So we are going to monitor the synthesis process of DNA by the DNA polymerase enzyme, and we are going to infer the sequence from that. In order for the synthesis process to start, we need some primers, right? So here we have two primers, RD1-SP and RD2-SP. Now RD1 is read 1 and RD2 is read 2. Now, obviously you're wondering that if we need only one primer to start the synthesis process, then why do we have two primers right here? So this answer I will not give you right now, but when I'm going to show you like read 1 sequencing and read 2 sequencing, then you probably will understand this process a bit better. Okay, so that is the library impression step. So this is done for every fragment that is there in the platform. So next, let's talk about the flow cell itself. So this is how the flow cell looks like. So you have 1, 2, 3, 4, 5, 6, 7, 8 lanes in this flow cell. And in each lane, you have plenty of these oligonucleotides, these small sequences. So I think if you are not colorblind, I hope not. So there are two kinds of sequences right here. One is the orange and one is the red. So the orange is basically the complementary of the P7 primer, which we saw in the previous slide, and the green is the complementary for the P5 primer. So these help the DNA fragments to attach. So for example, we have one DNA fragment strand right here. One of its ends attached to the green complementary primer, which is the complementary of P5, and the other one attached to the P7 complementary primer in the flow cell. You are wondering that why do we have only one strand right here? Because clearly fragments are double-stranded. The double-stranded DNA fragments into single strand, and then you need to put them on the Illumina sequencing flow cell. So the thing is, on the Illumina sequencing flow cell, these fragments will be quite sparsed around. So there will be one fragment here, then maybe another fragment here, another fragment maybe way back at there, and so on and so forth, because there are plenty of these, what should I say, complementary primers attached on this flow cell much more than necessary. When these strands randomly are dropped in the flow cell, they randomly bind in different distant places. And as I said before, since these are single stranded after DNA sharing, both forward and reverse strand, this kind of bridge will be formed by both forward and reverse strands in different parts of this flow cell. So next part is cluster generation. Now, the first question you may ask is why do we need to generate cluster? So understand that, as I said before, that I will start from here, but as I said before, you have these single strands for each fragment, like sparsed, and they are in different places of the flow cell. So it's very hard to monitor these standalone single strands, the synthesis process of these strands, and to infer the sequence. So what we try to do is we try to form a cluster of these. So, in the previous slide, if this is a single strand, then we want to actually make many, many copies of the single strand in the same place, in very close proximity. So now if we monitor the synthesis process of all of these together, it will be much more enriched and it will give us a much higher resolution in monitoring how the synthesis process is going on. So in the cluster generation, the first step is to use the DNA polymerase enzyme to create the complementary strand. And then if we denature, then two forward and reverse complementary strand is going to be created. And then again, there will be this bridge structure. And again, we will have DNA polymerase enzyme creating 4 strands from one. Then again, if we continue on this process for multiple cycles, it will have a nice cluster of this fragment. So the single cluster is for one strand of one fragment. So this is a very poor picture, but again, it drives the idea home. So if you have only 5 strands of 5 fragments, then there will be 5 clusters like this, right? In each cluster, there will be same copy of the same strand multiple times. So this is the process of cluster generation. Now understand that we are going to try and read the strand from here. This will be one strand of one fragment. We are again going to try to read this one strand from here, one strand from here. So if we analyze these clusters, we are going to ultimately come up with 5 read of 5 strands that are available on the flow cell. But on the flow cell, normally there are thousands and millions of these clusters. So we are going to read them accordingly. So now that we have generated the clusters, we want to know the sequencing process for read 1. So now we could have called it forward strand or forward reads, but we don't want to call that because we don't exactly know whether which strands are read 1, which strands are forward reads, and which strand reverse reads. We just know that, okay, forward or reverse reads all will be attached to P5 location. So let me just show you this image. So we can assume that everything attached to this P7, the orange locations, are either forward or reverse. So everything attached to the orange places can be forward strands, or everything attached to the orange spaces are reverse strand. So there is no mix in the orange ones, right? And in the green ones, it's the opposite one. Now, since I've said that in the cluster, our goal is to keep similar sequences together so that the sequencing process is accurate and is hassle-free. So the first thing we do is we cleave away all the P5 attached strands. So now we have only one kind of strand, so either forward or reverse. Then we start the sequencing process. So let's just talk about one of these, right? There are plenty of these. So let's just talk about one of these. Let's take one strand. The first thing we do is we provide the complementary sequence, which is the primer for the DNA polymerase. And this sequence is for the read 1 synthesis primer. So we looked at this in the library prep step, which is the innermost 2 primers. So we have these bases floating around here, ATCG, and we see that a fluorescent molecule is also attached to each of these bases. So red one for T, blue one for A, yellow one for C, and green one for G, right? So these are floating around. So DNA polymerase will start, and I'm just assuming here, maybe it's putting a T right here because maybe here you have an A. So the complementary of A is T, so it's putting it here. Then we judge what is the wavelength of this fluorescent molecule, and maybe it is similar to the red wavelength. So we deduce that it is red, or it is basically a synthesis of T. That means the main base is actually A, right? Now understand that this base that we just inferred, which is basically T, the synthesis is T and the base is A, this is for only one strand of this one cluster. So all of these will ideally form the red wavelength. There will be some error but majority of these will be red. So there will be a large red signal from this single cluster. And so if we have 5 clusters for 5 strands, there can be 5 different colors, lit up. Now, obviously, we don't have 5 different colors, we have 4. So a combination of these 4 different colors in 5 places, maybe red, blue, yellow, green. So let's actually see what it looks like in this picture. It's a beautiful picture. So every light that you can see here, separated light point, is actually coming from one cluster. So you can see that the machine can now understand exactly which places which light is coming from, and it will assign the, what should I say, base call accordingly. So as you can see, if we have 1,000 fragments in the flow cell, we have successfully read the first base of all the, same oriented strands of all those strands, basically. So it's a massively parallel sequencing process. So we have basically read all the strands, one base of all the strands at once in one go. So we have to keep doing this until we reach the end of the strands. So we have to loop over this. We have to keep doing this. And after finishing, for this particular strand, maybe this will be the sequence of complementary bases. So we can easily infer this particular original strand, and these will be the light up that will happen gradually in the cycle. So this is how we actually successfully read out the read one, right? The forward or the reverse. We don't know which one, but either one of these. So how exactly do we do this for read? So let's look at that. That is the reverse read. So not that complicated. So basically we have to get these P5 strands again, right? Because we previously clipped them off. So how do we get them back? So read to sequencing, the first thing we need to do is we obviously read our strands in the read one sequencing, and it was done by synthesis, right? Because we synthesized the complementary strand. So first we need to rip off these synthesized strands from all the strands, original strands. So afterwards, we can again, from a bridge structure, we can do one step of DNA synthesis. We can denature them to single strands. And obviously, right now, we get back the P5. So all the P5s are obtained now, and we need to cleave off and destroy all the P7s, right? So it's the opposite procedure. And then I think you know what to do. Same fluorescent image capture process on read two strand clusters will also be done. So the reverse read sequencing is now done. So let's now understand what is the benefit here. So we have successfully sequenced the forward part and the reverse part of each fragment. So this is called paired end sequencing. So let me clarify what this means. So if this is the original fragment, the double-stranded fragment, right? So maybe the forward strand is the orange one and the reverse strand is the green one. So we have successfully sequenced the forward strands, forward part, the 5' to 3' part, and also the reverse strands forward part, which is 5' to 3' strand. So now if we map them to the reference genome, we have to do two checks. One is whether the forward and the reverse part is intact in similar place. So if that check successfully satisfies, then we can say that, hey, this is basically mapped to this particular position of the reference genome. So we now know with high confidence that this particular fragment is supposed to map maybe in chromosome 1, position 1000, I don't know. Okay. One last thing I want to mention is Illumina sequencing, as I said before, mostly works with shorter DNA fragments. Normally cannot work with longer DNA fragments. Why is that? There are two reasons for that. Number one is you can see that it needs to do bridge amplification, and forming bridge with a longer fragment, for example, say 1000 base pair fragment, is not easy. So that process is not efficient enough. So the clusters are very small and the sequencing doesn't work so well with that kind of small cluster. Also, sometimes, as I said before, in Illumina sequencing, you need to apply PCR amplification in order to amplify your input fragment number or input DNA. And PCR amplification is very much biased towards smaller size fragments. It is because it is much easier for the DNA polymerase enzyme to recreate those smaller fragments. So that creates a lot of bias. And so because of this kind of biases, it does not become feasible for Illumina sequencing to work with shorter and longer fragments. So if you want to do longer fragments, you need to do something like SMRT alpha sequencing, which I have a video of already, link in the description. And also we can do also nanopore sequencing. The link of that is also in the description. So just to summarize, so the Illumina sequencing is a sequencing by synthesis process, because we are synthesizing the complementary strand using DNA polymerase enzyme, and we are trying to see what are those bases in order to read the sequence. Then it is massively parallel, as I said before, those lights. So all the fragments are sequenced together parallelly, and mostly short fragments of DNA are actually sequenced here. Accuracy is very high, and this is the most cost-effective and most widely used next-gen sequencing method. So that is all for this tutorial. I hope that this was one of the clearest explanation of Illumina sequencing. If you find the video useful, feel free to share it, subscribe to our channel, and also feel free to donate to our channel via the link in the description. Thanks very much.